Welcome to our next video. This is the second part of a two-part series about signaling proteins. Our goal now is to look at signal transduction of dimeric membrane receptors. Let's take a look. This is a slide from our last video. We talked about having three major classes of membrane receptors. Whoops, pin. We talked about 7-TM receptors in the last video, and this time we're going to look at two classes of dimeric receptors. And the first one we're going to look at is one that recruits protein kinases. So remember, kinases phosphorylate molecules. So let's take a look at this. And we know phosphorylation is kind of the on-off switch of many of these regulatory mechanisms. Okay, an example of this one is human growth hormone receptor. So if we look here, we're talking about receptor dimerization, so coming together as a dimer from the binding of a human growth hormone. So we have two monomer units. Each exists as a monomer, and each has multiple domains. So the pink is our extracellular domain. The orange is an intracellular domain. And then we've got this domain that spans the lipid layer. And in this case, that's a 1-alpha helix unit that spans that lipid layer. So now, as that human growth hormone comes into the picture, it binds to one of these. And when it does that, it causes these two to come together as a dimer. So we get this dimerized receptor right here. And while this cartoon picture shows it really just being associated at the top, what really happens is that there's a tight association down the whole length. It brings the whole thing together. So let's look at how that might work here. So if I look, here was my monomer unit right here, and here's the other. So these are my receptor with their extracellular and intracellular regions. Okay. And then I have this kinase that associates, is attracted to, and stays close to the intracellular piece of my receptor. So there's one of those on each. So this JAK2, it stands for Janus kinase 2. You don't need to know that. That's just what it is. And so we have these receptors. We have this hormone-induced dimerization. So again, they bond to some hormone right here, and it pulls these two together. And what you want to notice here is that as these come together, it brings those kinases together. This particular kinase, this JAK2 kinase, is a kinase that supports phosphorylation of hydroxyl groups. So we're going to phosphorylate hydroxyl groups on tyrosine. Whoops, not one, on tyrosine. On those tyrosine side chains. So what we see is that when we pull these two together in close proximity, we get what we call cross-phosphorylation right here. Okay, so the one of these can cause the other one to be phosphorylated, so those hydroxyl groups on tyrosine will gain phosphate groups. And then this one will do the same for the other. So we get these phosphate groups now bound to where we had tyrosine. Okay, and when they're phosphorylated, they're activated. And so now they can phosphorylate other important enzymes in the cell. And we don't know specific ones right now. This could be a lot of different things. Gene expression enzymes would be one example, right? But now that these are activated kinases, they can take ATP and break it apart into ADP and the phosphate, where that phosphate is going to be bonded to some molecule in the cell that turns on off some other regulatory pathway. All right, so that was an example of a dimeric receptor that recruits protein kinases, but some of these receptors are also in themselves protein kinases. So we're going to look at an example, actually two examples. We'll look at this epidermal growth factor receptor and the insulin receptor. So let's start with the epidermal growth factor. The key thing here, y'all, the big picture, is really that as these signaling molecules, as these primary signaling molecules bond to the receptor, it induces some conformation change, a shape change, that then causes some reaction or some further conformation change that causes an association. So we're going to follow that through these steps as we're looking at this. So when this epidermal growth factor bonds right here to the receptor, it again pulls these closer together, right? So when it's bonded, it's pulled these two monomers close together. Now in this case, the extracellular portion right here, so extracellular on the outside of the cell, is where the EGF binds. The intracellular portion, intracellular, in this case is actually also a kinase. So this is the enzyme that can cause phosphorylation. So the receptor and the kinase are on the same molecule. And when these things come together, when our two units come together, it brings those kinases together. And just like last time, when that happens, we're going to get cross-phosphorylation. Phosphorylation. There we go. I can spell. So cross-phosphorylation. Okay, so the domain on the left will catalyze phosphorylation on the right and vice versa. So now what happens is, take a look right here at this little phosphate group. It turns out that that provides a binding site for this small GRB2 protein. Okay? And when that happens, that sets up a conformation change in this little protein that makes it an ideal binding area for this SOS protein. So remember, all these proteins are tightly associated with the membrane, so they can move around on the membrane, but they're not generally allowed to just float off into the cell, right? They're associated with the membrane, which keeps them in closer proximity. So the phosphorylation provided a docking site for this GRB2, which induced a change in the conformation and the shape that makes it an ideal binding site for this SOS protein. And then what we see is that that SOS protein undergoes a conformation change to allow this RAS protein to bond. Okay, so the RAS protein right here is a type of G protein. So remember our G proteins from the 7-TM receptors. It's a type of G protein, and just like the other ones, when it bonds when it's in the GDP form, okay, when it bonds GDP, it is inactive, and when it bonds GTP, it's activated. So what we see is that when the RAS protein in its inactive form bonds the SOS, or closely associates with it, it causes a conformation change. And just like before, this molecule will kind of open up that active site. GDP can float out, and while it's open, GTP can float in. And so now we have the same RAS protein shown in the purple, but now it includes this GTP molecule, so it's the activated site. Once it's activated, it can lead to more signal transduction in various metabolic pathways. So the key is we're going to activate this, and now depending what type of cell we're in, this can cause other metabolic pathways to be activated. Let's talk a little bit about that RAS protein. So when we talk about this particular protein, okay, it's a key component in many signal transductions. So remember, signal transduction, we're moving the signal from one molecule to the next. Okay, it's a member of a class called small g proteins, or small GTPases. And they're called small because they're monomeric. Remember, the G proteins we looked at in the last video were trimeric. All right, so members of the family control a variety of cellular processes. And again, it is active when it's bound to GTP, inactive when bound to GDP. And just like before, it has intrinsic GTP-ACE activity, meaning that it can revert back to the inactive form on its own. It has its own enzyme that will cut off that phosphate group and go back to GDP. So that kind of is a self-limiting factor. And other proteins can also modulate that activity, so make it more likely to happen, less likely, faster, slower. Okay, so that was the epidermal growth factor. Let's look at a different example of insulin, because insulin is an incredibly important piece of regulating our metabolism, and so we're going to look at this. Polypeptide hormone insulin is secreted when the blood is rich in glucose. So essentially, insulin is this biochemical signal to say we've been fed. Okay, it exists of two polypeptide chains that are linked by disulfide bonds. The insulin receptor is a receptor tyrosine kinase. So again, it's going to phosphorylate those tyrosine hydroxyl groups, like the earlier one we looked at. A difference in this versus the last one is that this one exists as a dimer even in the absence of the insulin. So it is a dimer from the start. Let's take a look at why. So as we look at these molecules, first of all, here's our insulin. It's got two polypeptide chains. Okay, and then on the right, we've got our cartoon of the insulin receptor. I wish we had used different colors here. So these strands are not these strands, okay? We just use blue and yellow for both. So as we look at our insulin receptor, we have two subunits, each of which has an alpha piece and a beta piece. So when I look over here, the one on the left, here is my alpha subunit and the beta subunit. And notice in this case, this beta subunit is the transmembrane part. The other one also has an alpha subunit and a beta subunit. And if you look right here, you'll see disulfide bonds. That's what those black lines are indicating, which are holding these two permanently together. So they stay as a unit, as a dimer, even when there's no insulin binding. Okay, we are going to step through this process step by step. So where you see the gray box right here, okay, that's the part we're focusing on. So let's take a look. We've got our extracellular side up here, so outside the cell. We've got our intracellular part. And when we bond insulin, so we see we have insulin in our active site. So when we bond that insulin, it pulls everything closer together, brings the conformation change that brings these kinase subunits together in space. And remember, kinase are going to phosphorylate. These are also going to phosphorylate those hydroxyl groups on tyrosine. And so once they're close together, we see that we do get all these phosphate groups added. So we get that cross-phosphorylation. Once that happens, what you see right here is that one of those phosphate groups can act as kind of a docking site for this IRS1 insulin receptor substrate 1 protein. You don't need to know the name, but you need to know that this phosphate group acts as a docking site for the IRS1 protein. Once it docks, that keeps this molecule close to one of these kinase phosphorylate, right? And so we get these extra phosphate groups added to that protein molecule. Those phosphate groups, okay, add a docking site essentially right here, okay, for this phosphoinositide 3 kinase. That's a mouthful, right? But for this other protein that is a kinase, so it can phosphorylate things also. And when this happens, when this thing docks, it causes a conformation change that activates this molecule, and it can phosphorylate things. So what it does is it actually phosphorylates one of these, one of the hydrophilic heads of our lipid bilayer. Okay, and it changes it. It's going to add a phosphate group. So it changes it from a structure that has 2 phosphate groups on that hydrophilic head. So over here we've got our R groups. There's the tails of that thing, right? And notice we've got 2 phosphate groups. And it uses an ATP to add an extra phosphate group. So we go from what we're going to call PIP2 to PIP3. Okay, that's going to change the structure and the dynamics of that lipid head because it now has an extra phosphate group. It's going to have different intermolecular forces. As that happens now, this molecule can bond to it. So PDK1, so PIP3 dependent protein kinase, yet another kinase, right? Here's our kinase cascade. So we've got yet another kinase that's bonding to this, and when it's bonded, it activates this kinase. So again, kinases phosphorylate things, and so as that happens, when it's activated, it phosphorylates yet another, y'all, kinase called AKT. Sometimes we call this one protein kinase B. So PKB, protein kinase B. When it becomes phosphorylated, it's activated. So over here on the right, this would be an activated molecule. This molecule, this one's interesting and different from our last ones because it is not associated with the membrane, so it's free to move about in the cytosol, so it can move throughout the cell. and it can phosphorylate other enzymes in the energy regulatory system. So one example, it could activate GLUT transporters to move to the membrane to allow more glucose in, something like that. Okay, there were a lot of steps. We looked at them here with the diagram. Let's talk them through one more time with words for those of you who kind of like the graphic piece. What would it be? A flowchart. So we have our insulin and insulin receptor. And when that happens, it pulls these close enough together that they can cross-phosphorylate. Okay, so we see the yellow here. I've tried to kind of color code. So cross-phosphorylate. When that happens, it provides this docking site so it can bond this IRS1 protein, and then that is now close enough to become phosphorylated. And notice the idea here of amplification, that this amplifies the signal. And the reason for that is that this, once it's activated, can phosphorylate many IRS, not just one, but many molecules. So having one insulin bound activates these that can then activate and phosphorylate many of these IRS1. Okay. We get this protein-protein interaction, these attractions between the proteins, okay, which then activates this protein to convert to phosphorylate the PIP2 to form the PIP3. Once we have that PIP3, now it can bond to this PIP-dependent protein kinase, okay, which then activates this other kinase. And notice that there are several amplification steps where this can happen more than once, and hence we can get an extra effect. We don't need a single insulin molecule for every single reaction we want to occur. All right, talking through it just with some more words. So we have insulin binding that causes a change in the quaternary structure, right? The structure down here that causes cross-phosphorylation of the two kinase domains, activating the kinase. The activated kinase of the insulin receptor, so right here, phosphorylates these insulin receptor substrates, shown in the orange. These phosphorylated IRSs are adapter proteins. They essentially convey the insulin signal on down the line. So now this molecule will bind and then convert this PIP2 to the PIP3. And now the PIP3 can bind to this next protein. Okay. And then at the end, this AKT can phosphorylate enzymes that control the trafficking of glucose transporters, which can increase glucose uptake by the cells, as well as controlling insulin or enzymes that convert glucose into glycogen. Now we still need a way to stop this process. So two things that can happen. Protein phosphatases. Okay, so kinase adds a phosphate group, so a phosphatase removes a phosphate group. So if we have protein phosphatases, they can remove phosphates from the activated proteins. And then that'll terminate that whole line of reactions. Lipid phosphatases can also have an effect because they can convert that PIP3 into the PIP2. So essentially, we're going to terminate this action by removing phosphates, either from those proteins or from the lipids. And just to kind of highlight, so we looked at two dimeric proteins, or dimeric membrane receptors, and they were similar in that they both have kinase activity. They can both phosphorylate once they bind the primary messenger. Okay, in the first case, the receptor recruits a kinase that is closely held in with those IMFs. In the second case, the intracellular part itself is a kinase. All right, hopefully after these last two videos, you have a better idea of what a chemical circuit is and how a message can be passed from one molecule to another to initiate or terminate various metabolic processes.